The goal of this experiment is to synthesize ethyl acetate from ethyl alcohol and acetic acid at various conditions. The synthesized ethyl acetate is analyzed using a gas chromatography. Temperature study and effect of catalyst will be evaluated. To synthesize ethyl acetate, ethyl alcohol is reacted with acetic acid with acid catalyst and heat. The catalyst in this experiment is Dow X50, which is a type of ion exchange resin. Let's review the mechanism of this reaction.
First, the acetic acid is protonated by the catalyst. The ethanol, as nucleophile, attack the protonated acetic acid. Next, the proton transferred in water is released. Finally, deprotonated and final product is formed.
In the experiment, 4 samples are prepared, 2 heating samples and 2 room temperature samples. The reaction is done with and without catalyst. The samples are heated for 60 minutes, while the room temperature samples are reacted for 1 week. The amount of ethyl acetate at the end of the reaction is determined using a gas chromatography. The heating samples are heated in a hot water bath with controlled temperature. To stop the reaction, the sample is later chilled in ice bath. A standard and each samples are run using the gas chromatography, and then the chromatograms are analyzed. This is an example on how to analyze a chromatogram using the Logger Pro. To determine the mole fraction of ethyl acetate, a series of calculations should be done. First, response factor is calculated to compensate the sensitivity of the detector. Then, the areas are corrected using the response factor values. Finally, the mole fraction is determined from the ratio of the area to the total of the areas. The equations for these calculations are summarized in these tables. You can find these equations in Isidore. Followings are the sample calculations for this experiment. To complete the post-lab assignment, use the given experiment data in Isidore.